So in previous sections we learnt about replication, okay, transcription and translation, and here we will see the regulation of gene expression. Okay, now regulation occur at different level in eukaryotes. It occurs first it can occur in the transcriptional level, that is formation of the primary transcript. It can occur at the processing level, that is regulation of splicing. It can occur at transport of messenger RNA from nucleus to the cytoplasm, or it can occur at the translational level. So you need to understand that regulation of gene expression is very important, and in eukaryotes it can occur at various levels. Okay, so it can occur at the transcriptional level, that is formation of the primary transcript. It can occur on the processing level, that is regulation of splicing. It can occur at the transport of messenger RNA from the nucleus to the cytoplasm, and the translation level. Now genes expressed to perform particular function. Example, in E. coli synthesizes beta galactosidase that will act on the lactose, and it will form glucose plus galactose, and this glucose and galactose will be utilized by the E. coli as a source of energy. Okay, so in E. coli specific genes are synthesized, and these genes will synthesize an enzyme, that is beta galactosidase, and this beta galactosidase will catalyze the lactose into glucose and galactose, and both these energy sources will be used for the synthesis of source of energy. If there is no lactose, then there is no synthesis of energy. Okay. Now metabolic, physiological or environmental conditions regulate the gene expression at various levels. In prokaryotes, the site of initiation of transcription is the site for control of gene expression. That is, regulatory proteins may act as positively or they can act as negatively. When they act positively, they are called activators, and when they act negatively, they are called repressors. Okay. A promoter region is regulated by interaction of regulatory protein and operators. Operator is present adjacent to the promoter. Okay. So metabolic, physiological or environmental conditions regulate gene expression. In prokaryotes, site of initiation of transcription is the site for control of gene expression. Regulatory proteins may act as positively, that is catalytic, or it can be the activator, or negatively, that is it can be the repressor, or the deactivator. Promoter region is regulated by interaction of regulatory protein and operator is present adjacent to the promoter. Now in this gene regulation we'll understand operon, that is the Lac Operon. Concept of gene regulation was given by Francois Jacob and Jacques Monod, which is called as Operon concept. They work with lactose or Lac Operon in prokaryotes, that is E. coli. The Lac Operon consists of polycistronic structural genes, and we also know that eukaryotes have monocistronic gene expression, but in prokaryotes we have polycistronic gene expression. Now they have three structural genes, that is, first one is the Z gene, which codes for beta galactosidase enzyme, which will form glucose plus galactose from lactose. The Y gene, which codes for beta galactosidase permease, which will help to permit the entry of lactose from outside to inside. And the A gene, it codes for trans-acetylase, and this trans-acetylase transfers an acetyl group from acetyl-CoA to beta galactosidase. So when we talk about Lac Operon, it has polycistronic structural genes, that is Z, Y and A. Z synthesizes beta galactosidase enzyme, Y synthesizes beta galactosidase permease, and A gene synthesizes trans-acetylase, and you have to remember the functions of all these three. Now the regulatory gene, that is the I gene, it synthesizes what? It synthesizes repressor, not the activator. It synthesizes repressor, which represses the gene activity. Operator will do what? Operator will control the structural genes, and its activity depends on the repressor. Now the promoter gene will lie left to the operator gene, and here the RNA polymerase will bind. Okay, as you can see in the diagram, first is the promoter, promoter region, then the I gene, regulatory gene, which will synthesize the repressor, then comes the operator where the RNA polymerase will bind, and Z, Y, and A are given, which are the three genes. Okay, see, these are the three structural genes, Z, Y, and A, which will do their function. Okay, and this is the I gene, which is synthesizing what? This is synthesizing the repressor. Okay, here the regulatory gene is synthesizing the repressor. This you have to remember. Now, what will happen in the absence of this lactose? Now, in the absence of lactose, the regulatory gene will produce the repressor. The repressor will bind to the operator region. The RNA polymerase will be unable to move, and no transcription or process will stop, or it will be off. See, in the absence of lactose, the I gene is synthesizing the repressor. Okay, this repressor of the messenger RNA. This repressor is binding to the operator region. So this RNA polymerase, RNA pol will not be able to bind here at the operator, and when it will not be able to bind here, the messenger RNA will not be formed for these three genes, and they will not be translated into particular enzymes that are proteins. Okay, so you have to remember that in the absence of lactose, regulatory gene produce repressor. Repressor will bind to the operator region. RNA polymerase will be unable to move, means it will be unable to move in this direction and transcribe the various genes, and no transcription or process, no transcription will occur, and the process will stop here itself. You can see here the repressor is synthesizing here. Okay, then the repressor is formed. This is binding to the operator, and due to this, the repressor binds to the operator region and prevents the RNA polymerase from transcribing the operon. Now, what will happen if lactose is present in the medium? Now, when lactose is present in the medium, the lactose will bind with the repressor. You have to remember that whenever the lactose will be present in the synthetic medium, the lactose will bind with the repressor and it will inactivate the repressor. Now, when inactive repressor will be formed, it will be unable to bind to the operator, and so the operator site will be there. Okay, it will be empty, and then the RNA polymerase will move and bind to the operator, and it will synthesize, or it will transcribe what? Beta galactosidase and other enzymes. Okay, see here in the diagram you can see that the repressor messenger RNA is formed and the repressor is formed. Okay, now this inducer is nothing but the lactose. Okay, now this inducer, that is the lactose, is binding with the repressor and making it what? Inactive. Now, when it is inactive, it will not be able to bind to the operator region. It will not be able to bind here, and due to this, the RNA messenger RNA will be formed. See here, Lac messenger RNA is formed, and it will be translated to form beta galactosidase permease and transacetylase. Okay, when this will, the transcription will be there, and then the translation will be there, and all the three enzymes which are very important for the synthesis, for the utilization of the lactose will be formed. Okay. So, in the presence of lactose, you have to remember that the repressor will bind. Okay, the repressor will bind with the lactose and inactivate the repressor. Now, the RNA can bind at the operator region and it can transcribe the polycistronic genes. Lactose is an inducer. Lactose induces the function of structural genes. It regulates switching on and off of the operon. Now, low level of expression of Lac operon has to be present in the cell all the time. Why? Because otherwise lactose cannot enter the cell. And when lactose will not be able to enter the cell, the regulation of the repressor, that is a negative regulation, because due to repressor there will be no synthesis of the beta galactosidase. So there is a need for low level of lactose present inside the cell so that it can bind to the repressor and inactivate the repressor so that RNA pol can bind with the operator, okay, and then it can transcribe all the Z, Y and A genes, okay. So this was all about the Lac Operon.